as your moderator.
Okay, welcome everybody. I hope you're enjoying this second day. It seems like it's super well attended and very exciting. So it's great to be here today. My name is Carla Rothlin. I'm a professor of pharmacology and immunobiology here at Yale University. I'm also the co-director of the Cancer Immunology program in our Yale Cancer Center. And it's my absolute pleasure to moderate this panel today on autoimmunity and oncology. I think it's going to be a very exciting panel with a lot of discussions. So maybe as we get started, we can have a round of introductions. And so maybe Mary Claire can start.
Hi. Hello, everybody. I'm Mary-Claire Peakman. I trained originally as a pharmacist. I have a PhD in pharmacology. I did 2 of my postdoctoral positions here at Yale, so it's lovely to be back. And since then, I spent 27 years at Pfizer, based in the Groton, Connecticut campus. Most of that time was spent in early discovery for small molecule therapeutics. I led a number of our biology platform groups that supported our small molecule programs from idea to development candidate. So think about things like reagent generation, transgenic models, high throughput screening, primary pharmacology, high throughput ADME, all of those spaces where we'd work in very close collaboration with our chemists, some of whom I know are sitting here today, to drive things to candidate. And then for the past 3.5 years, I have been a partner on the Pfizer Ventures team. The teams that I have been responsible for were involved in the discoveries of abrocitinib, ritlecitinib, and Paxlovid. So it's been a very exciting time.
Very exciting, very busy time. That's wonderful, Mary Claire. Great to have you here. Asif?
Good afternoon, everyone. My name is Asif Khan. I'm the Global Medical Head at Sanofi for respiratory and gastroenterology therapeutic areas. A lot of the focus I have in my day-to-day work is on early assets. When I say early assets, these are treatment options currently in development, so phase 2, phase 3. None of them are yet submitted or approved, which means a lot of our focus is in understanding the disease, is understanding the patients. My interest has been a lot more in using AI technologies from quite some years before it became a buzzword, as well as patient centricity in clinical trials. That's what I did. I'm a physician by training, but I did a PhD in patient centricity within clinical trials. And happy to be with this fantastic panel and all of you here.
Thank you. We have another Carla here.
This is very strange to have 2 Carlas. It's my absolute pleasure to join today at very last minute, so please bear with me as I work through the questions. But absolutely delighted to be here. So I'm representing Merck Oncology Search and Evaluation. I've been with Merck for about the last 1.5 years, supporting everything in the early space until clinical proof of concept in terms of areas of search. We search in an agnostic fashion, agnostic of disease, agnostic of modality. We're very much interested in very cool science. My cool science story was having met Ranjeet Bindra and heard the Modify Bio story, and that was an acquisition that I led last year. So very excited to be here. I think the ecosystem here is fantastic, and so excited to hear from everybody that's got some cool science to share.
Thank you, Carla, for joining us today. And Sarah, or other speaker?
Sarah Klimkowski. I'm an SVP on our research and investments team at Royalty Pharma. So I sort of exist on the furthest end of the spectrum. We make investments in therapies that are post proof of concept, but we have done a lot of deals, and I have been at Royalty for almost 13 years now and done almost $12 billion worth of deals, and it ranges from sort of, like you said, therapeutic area agnostic. So as a team, we do investments all across the spectrum of therapeutic areas. And we also have kind of a range of counterparties. So traditionally, we've done deals with academic institutions. We bought a prostate cancer drug, a royalty from UCLA for more than $1 billion. But now we do a lot of different things. We do phase 3 funding for large pharma. And we also create synthetic royalties by being an alternative funding source for biotech companies. So instead of converts or equity or even debt, royalty can be sort of a drug-specific type of financing that can be really helpful, especially in these choppy equity markets.
That's wonderful. Well, great to have you all here. We will start with some questions, and of course, if you have questions later, we will open this to all of you. So we'll hopefully have a very nice discussion here. So from an immunology perspective, this is very interesting when we think about autoimmunity and oncology, because of course, in autoimmunity, we want to suppress this aberrant immune response. When we think about oncology and we think about cancer immunology and oncology, we want to lift those barriers and enhance the immune response against the tumor. So very interesting perspectives, seems to be two sides of the same coin. As we go into what are the potential targets, there seems to be some overlap. So maybe let's start with the basics. Let's think about targets. How do we think about the targets? Is there any scope to sometimes think about the same target that will be applicable in different diseases, from the autoimmunity to cancer?
Yeah, it's great starting point. So maybe I'll start by saying that we're a strategic investment fund. So primarily we're looking for innovative science that is really adjacent to the areas that we're focused on in our therapeutic area research units, that sort of aligns with our broad strategy within each of those indications. So that's sort of our primary goal. We're looking for things that could potentially be future impact on the Pfizer pipeline, could be potentially a future business development deal. Now, I would say our secondary goal still is that financial return. That helps us give a very positive internal narrative so we can continue making those strategic investments. And I would say that primarily what we do first is look at the lead assets for the companies that are coming forward to us. So we're very focused on first whether there's sort of some innovative science there. But then generally our sweet spot, I would say, is sort of Series A, Series B. And so primarily those are assets that are really approaching development candidate or sort of pre-IND. So when companies are in that stage, usually it's their lead asset that they're building the financing round around. And so it's that asset that we will really do the deep dive on, do full diligence on, and it's that therapeutic area that I would go to for internal championship. And so then I would ask that CSO and invite them to be on our investment committee as we make an approval in that investment. So usually at that stage, the second program is a little bit further behind. They don't usually have two assets, specifically not in two separate disease areas or therapeutic areas. So I don't usually go to two therapeutic area heads to get buy-in for the investment. But I will take the biology to that other team. So let's say it was a company that has a lead asset in oncology and they're sort of expanding into the autoimmune space. I would generally go to the autoimmune folks and sort of discuss at a high level the biology rationale, the mechanism, and see whether that would be remotely on strategy. So that helps me build the narrative internally for that strategic alignment, but it won't necessarily drive the decision. So I would say with the companies that I've had experience in, and I'm always interested in hearing different experiences around the room, but generally there has been a lead program that's moved forward in a specific therapeutic area. And often now, as you say, we're seeing companies expand either from oncology into autoimmune or vice versa. And they tend to do that for a couple of different reasons. So it's usually driven around the biology or the mechanism that they're working on, or the modality. So, for example, in our portfolio, we have a company, Therapeutics. It's working on the LRB2 sort of receptor signaling pathway. And as you said, that's exactly right. In one instance, it's a negative sort of signaling pathway that in oncology you would want to block to try and increase the immune response. And in the autoimmune space, you would want to go in the other direction. And so you do have different assets in those two different indications. So very much capitalizing on the expertise within the company, the biology and the knowledge of the pathway. So there, it's relatively easy. You take two different assets forward for the different therapeutic areas. We also have sort of modality plays like in vivo CAR T, or so we have Capstan Therapeutics, Interius in our portfolio, or something like Crossbow Therapeutics, where they're making T-cell engagers with T-cell receptor mimics. And I think when you capitalize on that modality experience, you still have the opportunity to play both in the oncology and the autoimmune space. But again, sometimes you're targeting a different cell population, or you have the opportunity to select a different target. So again, even in those companies, the assets tend to be different in the two different therapeutic areas.
Right.
In one company that's in my personal portfolio, I'm quite excited around, it's called Grey Wolf Therapeutics. It's interesting because they're using the same mechanism of action in the same directionality for both oncology and autoimmune. So they're working, I know, it is. And so this sort of leads into that question of can you have the same asset or the same molecules in different therapeutic areas. So in this particular instance, they're making small molecule inhibitors of ERAP1 enzymes, which are aminopeptidases in the endoplasmic reticulum that trim peptides for expression on the MHC complex. So their goal is to develop inhibitors that will change the antigen repertoire expressed by the cells, or presented by the cells. So in the tumor space, you want to modulate the antigen repertoire, stimulate new immune responses, and potentially save your exhaustion of your T cells by downregulating antigens that they're already responding to. In the autoimmune space, there are specific peptide MHC complexes that are known to drive the autoimmune diseases, and there's really strong genetic correlations in things like ankylosing spondylitis. And in that particular disease, it's very well known that ERAP1 is responsible for generating that antigen peptide MHC complex. So again, if you could inhibit the ERAP1, you change that antigen repertoire and you remove the driver for the inflammation. So again, so it's same direction, same mechanism of action. So if you start to ask about whether you could use the same molecules or the same sort of direction, mechanism of action in two different therapeutic areas, I would say if I mention that, and you can tell me, if I mention that to my BD colleagues or my commercial colleagues, they tend to start to hyperventilate right away. And it's usually a commercial sort of aspect that concerns them. And the typical case study they spout is the Avastin-Lucentis controversy. I see some people nodding, and I'm sure there are people in this room who know more about this than me. But I'll sort of stick at the high level and feel free to add any extra details. So Avastin was approved in 2004 for metastatic colorectal cancer. 2006, Lucentis was approved as an injection for into the eye for age-related macular degeneration. Mm-hmm. Both anti-VEGF biologicals from Genentech, right? And so what happened when the community was waiting for the Lucentis to be approved, they took the Avastin, reformulated it through compounding pharmacies, injected it into the eye, and it worked, right? Now, here you get into the commercial aspect. The cost of that reformulated Avastin to inject into the eye, $40 to $50 a shot. The cost for the Lucentis, over $2,000 a shot, right? So now you've got these two different, and they are actually different molecules. They're anti-VEGF. And, the rationale from Genentech is you would use Lucentis for the eye because it's an antibody fragment, and so the penetration into the retina is much better. But when you've got that commercial sort of challenge with patient access of $40 to $50 a shot versus over $2,000, you run into this sort of challenge that I think has sort of been controversial for a number of years. And this is where sort of I probably haven't followed the literature well enough. I know they're doing trials, but there's an ongoing controversy, to my knowledge, around which is more efficacious and what are the safety profiles. And do you really get higher efficacy and greater safety using Lucentis in the macular degeneration? So we get back to Grey Wolf, sorry, and then I'll hand on to somebody else. You get back to Grey Wolf, you've got two mechanisms in the same direction going into oncology and autoimmune. And so our rationale is it's a small molecule approach. We've been fortunate enough to choose completely different chemical series, different physicochemical properties, different PK, potentially different tolerability. But in the end, we're going to have that conversation. From an investor perspective, I would say we've got strong biology rationale for efficacy. We're going to go into the clinic in both indications. If we're lucky enough to see efficacy in both sort of areas, then I think we'll have to layer on the complexity of the commercial.
Okay, all the best for that. That sounds exciting. Thank you for sharing all this. Obviously, I guess this really extraordinary area of research provides a lot of opportunities, but with that comes the challenges, right? Still, you need to focus, but you might have opportunities for developments that seem to be quite challenging on how you make the decisions. Very interesting for me.
It
So,
definitely
please,
is. Can
Carla,
I chime? Yeah, I was
please,
going to say, can
tell
I
us
chime
about-
in? Because it's very true that we do hyperventilate quite often when it's like, oh no, is this an immunology or an oncology play? And I can certainly say, from the Merck perspective, so we work by therapeutic areas. So I'm covering oncology, and then my immunology counterpart sits across the hallway from me. And we spend a lot of time talking back and forth about mechanisms where it could apply to both, right? And I mean, we have an interesting example of an acquisition that we had done last year with Curon out of China, where they had developed a CD3, CD19 bispecific antibody. They had clinical data in oncology, so in AML and in NHL. And we were very interested in the possibility of that therapeutic for B-cell malignancies and autoimmunity, right? And so that's an example of at our board levels, we were actually having discussions about how we wanted to make this play in autoimmunity based off the fact that they had generated the data in oncology and had very promising looking data. So it's quite interesting. It's not often that you find mechanisms where they're both going in the same direction, where it's both oncology and immunology related, but it does happen sometimes. But that being said, also from an early platform perspective, we have a lot of discussions with companies that are now working especially on transcription factors, where there's a lot of discussion of how a platform can have applications in both the immunology and on the oncology side, where it may actually not be the target itself that has that play, but it's really the company as a whole. They've developed a platform that is very unique and very interesting and has applications across a broad number of therapeutic areas. They're very much similar to the way that Mary Claire described it, is that we spend a lot of time aligning our therapeutic area heads as to what is the opportunity here. And then typically we try to have a champion. There always needs to be a champion who wants to drive and stand up for the deal that we would potentially want to do. But this is something that we're seeing more and more, is this cross-fertilization internally within our organizations to build deals that are centered around multiple therapeutic areas. So
very interesting.
To add that.
So many potential applications, right? Really exciting, expanding time in this space. I hear from both of you so many potentials, probably a lot of competition. How do the companies that you work with try to differentiate themselves in this very competitive landscape? Maybe Carla, you can continue if you wish, but yes.
Sure. I happily can. I mean, the reality is that there's always competition, right? And we're always trying to understand how do we differentiate ourselves, but ultimately, how can we do the best for the patients? And I think that's what we're always, if we're trying to keep our eye on a goal, it's how are we going to make an impact for our patients, and how are we going to do it better than our peers? And that's truly the nature of the conversation, right? It is absolutely challenging, but the reality of it is that the probability of success of our therapies making it to patients is the reality of our industry, and it's going to take many shots on goal before somebody has the exact right approach to get that therapy to patients. So as much as we'd like to say that there's a lot of discussion around what the competition is doing, we really try not to distract ourselves with it, because the reality is that they may either succeed or we may succeed, but we all need to keep pushing forward.
That's great. So commitment to what you have chosen. Sarah is...
Oh yeah, I was just going to say, we have such a different perspective, but as I thought about this panel, and I also sort of joined last minute in terms of first panic, and then just thinking about how these two spaces have really interacted with in the past. And I was surprised following Chemo how many examples there are in oncology that pre-lead development in autoimmunity. You have CD20 with rituximab, which obviously led to ocrelizumab, which is sort of an incredible therapy in multiple sclerosis, same mechanism, but to the point of sort of oncology leading the way, and that natural mechanism of NHL and B-cell malignancies sort of portending, development in autoimmunity, BTKs, CD19, BCMA, CD38, So I was just thinking about, once they get to where, post proof of concept, so once they get to almost like pre-approval stance, you think, what makes it good for autoimmune versus great for oncology? And obviously you have different aspects of that. And a lot of it is also sort of about your patient, and as you think about just, the sort of the OG, as we were saying, rituximab versus ocrelizumab, where one is for, an NHL or a CLL, and one is for multiple sclerosis, and these patients are on maybe CD20 for 10 years. Does dosing matter? Side effects obviously matter. So how do you choose a bespoke therapy for sort of the right patient populations, even though a lot of the biology is sort of overlapping in interesting ways? Also, BTKs, if you think about treating in CLL versus needing something for MS that's CNS penetrant. How do you choose the different molecules that are sort of better suited for these different patient populations? So I just think that that's kind of an interesting way to sort of view it, because it's such a well-trodden space. Maybe from a commercial standpoint, it's challenging because these are such different populations. But from sort of a fundamental biology standpoint, there's so much overlap.
Yes, thank you for bringing those beautiful examples.
Thinking about assets, right, when you get more late, right, into development phase 2 and phase 3, a key way to differentiate is thinking about the patients, right? What outcomes are you improving versus whatever other treatment option is available? I'm from medical. I don't think of them as competition. I think of them as other treatment options for our patients. How do you differentiate that? If you don't, forget about it. The other way is, what patient population are they targeting? Are you targeting? Are you bringing a solution for patients who have no other solution? Right? So think about the outcomes, thinking about those subgroup of patients. Maybe there's a different phenotype, maybe there's a different endotype. What has happened often in immunology is an unmet need was identified in a certain severe subpopulation, and almost all development went towards that, right? That's the only patient population with unmet need. But as we look more into diseases, we see there's an unmet need across spectrum of severity in diseases, which means there are more patients who need treatment options, but nobody has really looked into that. I've worked in some therapeutic areas or treatment options where I had payers as well as policymakers say, well, this is not a disease, it's a nuisance. Why are you even thinking about this disease or a treatment option for this disease? They have sufficient treatment options available. When we're able to generate evidence to show how much their lives are impacted, that condition, patients had worse life than a patient who had severe rheumatoid arthritis, for example, or had a heart failure, but it was not visible. Right? If you think about heart attack, that's like, okay, that's bad. But if you think about a patient having runny nose every day, what do you think? It should be okay. Any treatment should be fine. But no, it's not, because there are the downstream effects of these symptoms for patients as well. So there's multiple ways. So it's outcomes, patient populations, and what you're bringing near for the patients as well as the providers.
That's wonderful. Thank you for bringing the patients as the key focus for everything you do, right, in the end. Maybe following up on the patients and thinking about them. We know, particularly in the autoimmune space, in many occasions in the past, we focus on the symptoms and how to alleviate those symptoms. There seems to be a lot of progress on trying to really change the course of the disease and disease-modifying agents. Can you comment on your experience about disease-modifying agents, and how do we look for them? Is there a way we can focus on advancing this type of treatments?
I won't get into translation medicine. That's not my expertise. We'll talk more from once you have an asset. One key thing, we can break it down into a few things. One key thing is the disease biology. You have to understand the disease biology, that there is a causal relation between your target and the intervention, not just an association, right? The other aspect would be this temporal aspect of it. Is it downstream or is it upstream? So when we speak about disease-modifying agents, they have to be a bit more upstream. And the reason for that is once you have a target, an intervention which is upstream, it can impact the progression of the disease. It can potentially delay it or even prevent it, which then brings us to one other interesting aspect, which is when do we need to provide, if you have an intervention you believe could prevent the progress of a disease, which patient do you give it to? In immunology, what has happened often is as disease progresses, damage is done throughout. And a lot of these interventions come in at that last stage for the patient when they're so severe that you can't reverse any of that. And the reason for a lot of that is excess issues, what payers and policymakers would think is a patient population with unmet need. So you may have a disease-modifying treatment, but you also have to think, in the lifetime of that patient, when do we give that intervention? Then how do you identify that patient, right? So a diagnostic biomarker, a prognostic biomarker, the field is really moving towards, and we are moving towards, looking at patients much earlier in their disease. So when you intervene, the change actually leads to either prevention of the progression of the disease, or it delays some of the worse outcomes. So you also definitely need biomarkers or surrogate biomarkers to look into that. However, developing that from a clinical trial perspective is complex, and it also brings in this aspect of what regulators want as an endpoint for approval. You would have to still do your traditional clinical trials to convince them. You may include some of these endpoints in maybe a follow-up phase 4 studies. You may want to study it through registries. But ultimately, what it should do is, if you improve your functional aspects, I'll give an example just after this, it should improve patient symptoms, how they feel, how they function, what's their quality of life. So regulators often focus on hard endpoints, functional endpoints. If you go and tell a regulator, for example, a symptom improvement, for some diseases, yes, but asthma, as an example, is a disease where you have this loss of lung function progressively over time. By the time a lot of these monoclonal antibodies have come into play, it's when the damage is done, right? So it's like those severe patients. So if you come in much earlier, you could potentially prevent that progression of declining lung function, or you could potentially even reverse it, but it's the time frame also when you come in, right? The other complexity then is, you may see in this data which has shown that if you improve lung function, doesn't correlate to symptoms for patients. So we have functional improvement, but you have no improvement in how the patient feels. So you have to, again, look at treatment options which can do all of that. It can't be just one. It has to mean something for the patient as much as for the lung function improvement. They won't feel it. But can they breathe better? Can they walk better? Can they sleep better? That's critical. That's what truly a disease-modifying treatment would be.
That's wonderful. Carla?
Yeah, I'd love to add something to that, because I know there's a lot of innovators sitting in the room here. And I hope what you're hearing is these are our aspirations in terms of how we want to see these drugs get to our patients. But the reality is that these conversations need to happen very, very early. So when you have those early ideas and you have these biological mechanisms that you're trying to go after, trying to understand these additional complexities of how are you going to choose the patients, what are going to be your PK-PD relationships that you need to establish in your preclinical models, the more of those pieces that you can tackle as you're starting to build your ideas, the more helpful it comes down the line that we don't have to solve this kind of late on in the game, right? Especially in the asthma space, as it's such a heterogeneous population of patients, you have to subtype these patients. If you can already start to build your biology around those pieces, it just saves so much time, and it makes it so much easier in the later stages of the drug development cycle to be able to do that. So I just want to throw that plug out there for anybody sitting in that really early space and thinking about your ideas, is start to bring in those conversations really early to the conversations that you are having of how you want to develop these therapies, because ultimately that's what makes the difference for us to be able to develop those to drugs that make an impact for patients.
If I may just add on that's a fantastic point, is the reason potentially all those treatment options were developed much later in the disease paradigm is because everybody saws that's where the unmet need is. Nobody has looked at, unmet need before that severe disease paradigm. And if you go and look into literature, there are disease areas where if you go into milder patient, you'll see very scarce literature because nobody's studying that. So you have to also consider that, right? Okay, these are the treatment options, but why do patients progress? If patient progress means that current treatment options don't work as well in all the patients, and you have to start identifying those patients, whatever those phenotypes or endotypes would be.
Wonderful. Thanks for highlighting the complexity once we get into the patients, right? Really wonderful discussion, interesting area with complex targets and, very interesting opportunities to advance this to the populations that many of you care. Now, a key aspect to really make a change is also to invest accordingly. And this is very important so that this gets to the patients. Let's change focus a little bit and talk about investment. what are the investment strategies that have really worked in your experience? I know maybe Sarah, you can start commenting about this. How do we really push this to make this a reality for the patients?
Yeah, obviously a critical question, because sometimes what we see as the hardest step is sort of that last step, actually getting it to patients, and not only that, but getting it to patients in an affordable way and a broad way. But the role of financing and sort of funding at that stage is, I do think it's changing, and I think that we see innovation in financing drug development as well, and very happy to be a part of that, because we see really exciting strategies, but companies at sort of the mercy of equity markets, and not often you see things shelved even at large pharma that could be really important for patients. So I guess overall it's sort of the breadth of funding options and sort of a healthy ecosystem of innovation. So folks who are interested in funding things that are early, mid-stage, later stage, post-commercialization, and sort of a lot of options. And I think, just having that innovative ecosystem, and academic institutions, tech transfer, being involved in all that all the way to the end. And royalty financing is part of that innovative ecosystem. Not really I think what we always say, and as we think about making sure that these, life-changing therapies actually reach patients, is that it's a complement to sort of more traditional options. But it has only been around for 15 years really for biotech companies as an option. So I hope that there are other interesting, innovative ways that we can sort of lean on in sort of a crunched funding environment that we're in now, that sort of get those dollars to companies in a sort of a diverse way, and sort of build out, whether it's therapeutic area or drug-specific financing, which is what we generally do when we work with the innovators, or whole financing. That's a start. Yeah.
Carla, sounds like...
Yeah, I think, it is a crunch time for funding, right? I mean, I think we'd all be remiss if we didn't acknowledge that in this room. But the reality is that I think for a lot of innovators, the one key message I always like to leave everybody with is that you need to be open-minded. You need to be very open-minded about how you think you want to get to that next stage of moving your asset along. And whether that's through non-dilutive funding, whether that's through a royalty stream, whether that's through, a venture arm, you have to be open-minded, and you have to talk to as many people as you possibly can, because there is always a path forward if you are open-minded. The worst conversations that I have on the day-to-day are the companies that come to me and they say to me, "We are not looking for a collaboration. We only are looking to be acquired." And I'm like, well, good luck. Good luck. Have a great time with that, because you know what? I may have no budget for an acquisition, but I may have tons of budget for a collaboration, and you just shot yourself in the foot. So thank you very much for coming out. So please don't do that. I think the ability to be open-minded and talk to as many of us as you can, because you never know who has a space in their portfolio, who has a hole they're trying to fill. It might be that, okay, it's very obvious to the outside world that Merck is not invested or swimming in the lane of RLTs, but does that mean I don't talk to RLT companies? No, not at all. I happily speak to anybody working in that space, because the pendulum is always shifting in our organizations. And while you think you might have a view from the outside of what we're working on and what we're interested in, you never know where we're actually planning to make that next investment. So I think that's just something that's very important for this audience, is to remember that.
Thanks for being so candid.
Yeah, I guess I'll echo everything that Carla and Sarah said. And I think one of the things I love about this ecosystem, and I do use that word a lot in this role, because it is such a networked group. And as you say, there are people at all sorts of different stages from an investment perspective. And we tend to get to know who's doing the seed and the pre-seed stage, because we want to talk to them as their companies are getting ready for the Series A. And who do we talk to bring into the Series B and the Series C? I think, as you said, being open to different types of partnering, and certainly within Pfizer, and I'm sure it's true with all of the other large pharma companies, when we meet with entrepreneurs and new startups, we generally try to find the right fit and the right type of collaboration that works for both the company and for us. So we very early on tend to have everybody in the room in those early conversations, whether it's people from the research side who might be interested in collaboration, somebody from the venture side, somebody from the BD side. And as we have those initial conversations, understanding where the company is, what they're looking for, we will tend to have the right person take the lead as the champion, whether it's someone from BD for a later stage asset or whether it's somebody on the ventures team for a ventures investment. But even as that relationship progresses, more things may come into play. So, as I take a role, we take very active roles in the companies we invest in. I usually try to take a director seat on the board. We usually are talking to the companies about what their BD strategy is, what their research collaboration strategy is. And this is not a competitive environment in a way, because we're all coming together as a syndicate. I co-invest with Merck in some of our companies, and all of the other corporate venture capital groups. And when we're in that role as a director, we're looking for what's most beneficial. Our fiduciary responsibility is to the company. And so we are very supportive of them taking on non-dilutive capital. That's great from an investor perspective. It's great from the company perspective. But whether there are BD deals that are available with other companies, we're very supportive of that. So that's one of the things I really like about this whole area, is that everybody's focused on getting something through to patients.
Wonderful. Thanks for sharing all that experience with regards to investment in this area. So I think we touch upon really interesting aspects with the targets that you're focusing on, the patients, which are our key focus, and how we invest appropriately. Now, of course, this is not done in isolation. We live in a world where there are many new developments. I can think about how AI is transforming our world, also data science, for instance. So let's change gears a little bit and discuss about how this is impacting what we do. Maybe Asif, I know you lead efforts in the AI space in your company. Can you tell us a little bit about how that new world is shaping your focus on identifying new drugs for unmet needs or trial design?
That's an interesting, and it's a buzzword these days, right? AI, and something to think about. I've led development of some platforms which help us analyze data much faster. We could go from a hypothesis to a final study in days, I would say, or even in minutes and seconds. We can do some really good analysis using a platform which uses these technologies. However, the reason for that was that we as companies have a lot of data, and you have to go to a specific function to request for analysis to understand that. But we sort of created a platform understanding some of the key questions are always the same. Can we automate that? And we were able to do that. However, for AI, one thing to remember is AI is only as good as the data you train it on. We often blame AI for coming up with the wrong answers. It's not the AI that's at fault, it's us who are at fault, because we're still feeding it the same data. We're doing it at speed, but it still has the data that's coming from whatever source. So the first thing to think about if you're using AI for any analytics, any population understanding, unmet need understanding, is to make sure you're getting the right data as well. If you're only getting data from, the hospitals associated with Yale, you're going to look at a certain outcome, you're saying it works very well, the intervention is perfect. If you didn't look into INSTI, for example, for that same intervention, you'll probably see very different outcomes. So getting the right data is really critical before you think about AI. And then you can use AI because it will give you the ability to execute at speed and at scale. But I do focus a lot on data because big data is another fancy buzzword. What I rather prefer is deep data. Data that has all the variables you need to answer the questions that you have. It's not the volume of data that matters. Clinical trials are not done on tens of thousands of patients. Some maybe, but not all. When you look at claim databases in the US, those are millions of patients. But the RCT data, which could be a few hundred patients, can get you a regulatory approval. 100 million patients can't get you a regulatory approval because it doesn't have the outcomes you're looking for. So when you're applying again AI, you have to make sure you have the right data to ask the question. And then one other key aspect is AI can understand patterns, it can give you insights which, normal or classic analysis can't. But AI does not have wisdom, it does not have empathy. You have to make sure that we don't get into artificial intelligence, but rather what I like calling augmented human intelligence. It should help you, but it should not take the decisions for you. Because one of the issues with AI is only because of the data, again, that you feed into it, it can actually augment bias. And you don't want that, right? It may be able to only identify unmet needs, which are there because of the bias in the data you collected. So there are those certain railguards you need to be fairly certain of when you're looking at AI. Many years ago, we did a study. We used natural language processing. So we had patients write down the answers about how they felt about a certain intervention. What was interesting is, depending on which country the answers came from, they could be 2 lines or they could be a full page. So when we used NLP on that, the interpretation by country was very different. But because we're humans, we could look at that and say that's because of cultural differences. So this may be good for country X, it's not good for country Y. Because if your answer was it was good, it was bad, what do you make out that? So we have to really think about AI. It's a fantastic tool to use. It can speed up, as I said, evidence generation, but we have to be very thoughtful in how we execute it.
Thank you. Yes, look, sounds like
I actually had a question because, I think there's been a spate of AI companies that are churning out particularly asthma and COPD sort of established target, but they use AI, and then I guess their goal is to identify a very, high probability of success asset and then take it very quickly into phase 3 trials. And I was curious, with your experience with AI and also with asthma and COPD and immunology, like how you think about that approach and speed to market is so important. Is that something that, other people's perspectives too, that sort of worth the risk benefit?
I would speak about using this technology to understand the patient populations, right? To understand those phenotypes or undertypes, which may have a higher unmet need. AI can definitely help with that. But does it get you a drug faster in research? Yes. You could do a lot more analysis much faster now with all the computational powers you have available. And you can even look at these big data or deep data much faster. So it should help you speed up. But it's not going to, again, replace some of those key requirements that we have.
Thank you so much. As somebody obsessed about data, I appreciate the focus on the discussion on deep data. I hear you there. Deep, rigorous, right? Important data that can really shape how you advance this area. This is wonderful. We have some more minutes left. Maybe before concluding, I would love to hear from each of you. What are you most excited about? Let's think about the next 5-10 years. What are you seeing coming up?
I think there are certain modalities that are sort of top of mind for us right now. I mean, Pfizer, probably well known for our ADC space right now. That's a space we're looking at, and degrader approaches. I mean, those are sort of modalities that have been very hot for a while and remain hot. I think we're seeing a lot of sort of multi-specific T-cell engagers or multi-specific antibodies. And I think what is sort of interesting to me is how we're understanding what multi-specific potentials to bring together, what targets to bring together. I mean, you just saw our sort of 3S bio. We're in sort of the PD-1 VEGF space lately, but whether you're thinking about sort of proximity proteins, protein stabilization we were talking about last night, what are the partner proteins that you need to bring together to, as you say, hit some of these multiple mechanisms that are going to be necessary to really drive disease into remission?
Can we really target that complexity in biology? And please feel free to answer it based on your experience. I know each of you bring a little bit of a different experience to this path, which is wonderful.
So I'll again focus back on patients, right? So what I'm excited about it, we have a lot of new treatment options, as many we are working on, but how do they actually change or break those ceilings of efficacy? Are they more safer? I want to see how these treatments change what we know today and what we can achieve today. Because eventually, at the end of the day, if patients don't feel better with any treatments, to me as a physician and somebody working in industry, the treatment is no good.
Thank you.
I think very similar to Marie-Claire, from the Merck perspective, we're obviously also heavily invested in ADC. So this is a space that we're super interested to see how we can make a bigger impact for patients. I think, seeing a lot of covalent chemistry companies, I'm really intrigued to see how covalency is going to play out, and how that's going to enable us to go after traditionally undruggable targets that everybody's been trying to go after as the holy grails of at least oncology. Think of beta-catenin and MYC as examples, and how we can go after these very different pockets on proteins and whether or not how that's going to play out. I think that's super interesting. I think the degrader space is, again, another area where I see a lot of companies coming to development candidate stage, and it's a challenging CMC path for a lot of those molecules, which I'm really hopeful that a lot of the big guys are going to get behind so that we can help support those companies, because some of the degrader molecules that we're seeing coming out of the preclinical space, these are absolutely mind-boggling what they're able to achieve. But it's going to have inherent challenges, and I think that that's part of the challenge of where we're sitting now and how we're going to be able to leverage the big organizations to be able to put muscle power behind that and make those assets a reality. I think the one other space that I'm very intrigued by as well is this whole idea of tissue targeting, and how can we get modalities to the tissue so that we can limit off-target and off-target toxicities or unwanted profiles of our molecules. So how are LNPs going to come into play for delivery of siRNAs as an example, right? I think we all thought that siRNAs were going to revolutionize drug discovery, and lo and behold, they have not yet. And I say yet because I think there's probably potential that they could at some point in future. So I think there's a lot of really cool ideas, and it's a matter of really trying to leverage everyone's expertise in the room to try to bring some of these ideas to reality.
Thank you for highlighting those. I think also as a basic scientist, I cannot help but say that how exciting to be here where, so close to us, all these degraders, were invented in a sense. So very exciting for me to remind us all about that. Sarah, some last words from you.
Yeah, I was just going to say, I think we've always been excited by, improvements in efficacy. That's kind of where we focus a lot of our time and energy and watching new data come out. But the other thing that more recently has been really interesting for me and something I'm focused on is sort of patient choice, because options are so effective that at some point it turns to sort of really patient-centric. What is the best fit for that particular patient? And patients are individuals and they're different. We just were at the cardiometabolic. There's so many options in the anti-obesity space, and more coming. What does the patient choose? And sort of patient choice can go into plenty of other, conditions and diseases as well. So that's something we're really focused on, is sort of trying to get more into the focus of what does the patient want? Because the options, you'll talk to docs and they'll say, well, all these options are effective. What does the patient want for their lifestyle? And I think that's a key part of how we think about the world right now. Particular.
Thank you for your efforts in thinking about that. I think we have all been patients maybe at some point, so we understand the complexity of that situation. Thank you to all our wonderful discussants today. I think we will remain here. Can we open it for some questions? If anybody has questions, okay. I think I can get close to you and we will bring you the microphone.
So first of all, this was fascinating. I've really enjoyed listening to all your thoughts. I have one thought, and I'm super excited about RNA and AI, and we developed an RNA AI platform for next generation cures. And we can imagine that we can bend the time and cost curve for new drug discovery and development and approvals, because one of the problems is still, when I started looking at genomics, it was 1 billion, 10 years, 5% success rate, and now it's 2 billion, 10 years, 5% success rate. So I think that the gap is similar to what Asif is talking about, finding the unmet need in the particular patient population, and that means that you need to do much more genomic testing and screening in order to understand where that is. So I would like somebody to comment on finding the right patients for your drugs using AI.
I'll go back to what I said earlier, right? Using AI, I'll give example of asthma, right? There are many biologics, monoclonal antibodies currently approved in that space, and most of them are only for patients. If you look at the treatment, the global treatment guidelines, they've got gene, and there's step 1, 2, 3, 4, and 5. So, as you go from mild to severe, you increase the treatment options, which again would tell you, if you look at it, is like only those on step 5 have unmet needs, right? Because that's where the most severe patients are. But what AI can help you with is, if you have data on all these patients, to understand which of those patients look similar to the ones on the step 5, the most severe ones. What are their characteristics? Which are the patients most likely to progress faster? Are there any prognostic markers you can look at? Are there patients who have certain other diseases, be that cardiometabolic, be that immunological diseases, who have the potential to progress faster, which means if you intervene earlier, you may stop that. And intervening earlier is always complex because you do need, again, long-term data. That's where I think predictive modeling from AI can definitely help.
Any other questions? Our discussions will stay around, so you can come up to them later and discuss. I want to thank each of you. Thank you so much for joining us, for sharing your thoughts. And thanks to all of you for being here and participating in this. Thank you.